Hello, my name is Anthony Gonzalez, and today I will be giving a lesson on a topic for biology. And the name of this lesson is Let's Get Sick. Well, imagine yourself walking down the street having a good time, except it's not a good time. You have a headache, you're wheezing, you're coughing, you're sneezing, and you have a pocket full of tissues. And you know those are clear signs that you're sick. Now, you're always sick. You're always sick. But do you ever take the time to really think, well, why exactly are you sick? Well, that is actually because there is currently an agent in your body. Now, when I say someone as agent, you probably think, oh, someone from an action movie, like 007, James Bond, obviously. Well, close, but what I'm talking about are viruses. Now, let's get into the real details for this. What is a virus? Well, a virus is defined as a small infectious agent that can only survive off of a living host. Now, what is the host usually for a virus? Now, the host could be an animal cell, like our cells, plant cell, or bacteria. And as you can currently see, one of the hosts it could be is a red blood cell. Now, a virus is also considered to be non-living. Now, what exactly does that mean? Well, what it means by non-living is that it does not have the necessary parts to be considered living. Now, the necessary parts for a cell to be considered living is it must have a cell membrane, cytoplasm, DNA or RNA, and ribosomes. And as you can see to the diagram to the right, you can see all those labels on an animal cell. Now, if you do not see DNA, that is actually included inside of the nucleus. Now, let's go on to the main virus that we will be looking at today, and that is a bacteriophage, or for short, phages. Now, what this bacteriophage is comprised of is DNA or RNA located inside that orange polygonal figure, which is actually called the capsid, which is a protein coat that goes around the DNA and holds it there until it is necessary to infect the host cell. Now, the capsid is comprised of tiny individual proteins called capsomers, which make up the capsid itself. Now, if you look all the way towards the bottom, you can see the base plate where all the little legs are, and that is lysozyme enzyme. And what that does is it speeds up the process in which a virus, or in this case our phage, can infect the host. Now we will go on to the first cycle in which a phage uses to infect other cells, or hosts in this case. Now the first cycle that we will be looking at is a fast and brutal cycle called the lytic cycle. Now the reason I call it this is because this cycle is basically for the virus to go in, kill hosts, and basically spread itself until it dies. Now what I mean by that is let's go through the steps on what happens in the lytic cycle. Now, what happens is first the virus attacks a host cell, then from the protein coat or the capsid it injects its viral DNA or nucleic acid into the host cell. There, in the host cell, it will start to replicate the virus DNA inside of the host cell. There, it will also start to create the necessary parts for a virus to be created, almost like the host is being a type of factory for the virus parts. And there, inside of the host, it will then begin to build more and more viruses, until for the last step, which it will then lyse, or in other words, or as you can see in the image, lyse, which means to cut its way out of the cell membrane so that it can repeat the process again. Now the virus assembly that happens inside of the host cell is the virus, as you can see, this is the virus assembly. What happens is the DNA mixes with the head assembly. It creates the DNA packaged inside the head. Now that is the main part, or the important part of the virus, because that contains the DNA necessary for it to spread. Now if you look to the left, it has the base plate, the tail core, and then the sheath added around it, which is just protein. Now this is basically so that the so that the protein can go
DNA.
through the head, through the base, go through the tail core, go out the base plate, and into the host cell itself. Now, as you can see to the bottom right, I did not talk about this too much, but those are tail fiber assembly. Now, what that is so it can attach itself to the host itself, and that is basically how it gets on. And then after you do that, after it's all completed and all put together, you have a mature phage, or in this case, virus. And then the next cycle that we would be talking about is a slow and everlasting cycle, and the reason I call it that is because this is a very hard virus to get rid of, because it basically goes through many cells, and it is basically just a large spread of virus that is just very hard to get rid of. Now, let me stop dilly-dallying and go to the lysogenic cycle. Now, what happens in this cycle is that our bacteriophage attaches itself to the host. It then injects its DNA or RNA, and then the DNA or RNA integrates with the bacteria's DNA. And then after it integrates with the bacteria's DNA, the bacteria will then begin to split off and create two more DNA. Will begin to split off and create two more bacteria. Now, when it does this, it will actually replicate the virus's DNA, and then you'll have two bacteria that has the virus, that has the phage DNA in it. Now, for this to actually work, the bacteriophage must be temperate, which means that it won't go straight to the lytic cycle, but instead will go through the lysogenic cycle. And once it actually bonds with or integrates with the bacteria's DNA, the DNA will then become prophage, meaning that it has the virus's DNA in it. And then it will, when it splits off into two more cells, it is then considered a lysogenic cell. Now, this will be a quick topic that I'll go over, and it's just a little extra. We're going to be talking about a retrovirus, which is just a different type of virus that actually, it is most common for using the lysogenic cycle because it uses RNA and the reverse transcriptase, and that basically makes the process of going through the lysogenic cycle much faster than the other bacteriophage. And as you can see, it does the same process, but it has it has a different type of look to it, such as having a lipid bilayer and a protein envelope, the capsid protein, and the RNA and reverse transcriptase, which will basically make the process much faster. Now this is also another extra. This is not an important part, but it's just to give you a feel of that all viruses don't just look like the ones that I presented on to you right now. Now, as you can see, for A is the heletical virus of rabies, B the segmented heletical virus of influenza. As you can see in there, you can see the DNA strand inside of the capsid. For D, it's an envelope isohedral herpes simplex virus. As you can see, it's towards the bottom of it, you could see where it would attach itself to the host, and the top where the capsid is, where it contains the DNA or RNA, and the E, the unenveloped polio virus, and the F, which is the isohedral human immunodeficiency virus with spikes on its envelope. Now, this is an important part. This is just to give you a feel of what the other viruses look like. Now, as you can see, all of these look very different, and the reason for this is because they all have different types of ways of infecting the host, and much like the base plate for our bacteriophage has a way of speeding up the process of spreading itself and conquering many cells. Thank you for listening to my lesson. I hope you learned something today. And don't get sick.